Hi class, this is your instructor, Skylar Huff, and we're now here in Chapter 11, The Basic Principles of Heredity. Human characters such as eye color and hair color, along with a multitude of other characteristics, are passed on from one generation to another. With that, I call heredity is nothing more than that transmission of genetic information from parent to offspring. Generally follows predictable patterns in organisms as diverse as humans, penguins, baker's yeasts, and sunflowers. So here I'll say genetics is that science of heredity, and of course it studies both the genetic similarities and genetic variability or genetic variation. So I call genetic variation the differences between parents and offsprings, or among individuals of a population. And of course, you could define a population being composed of groups of individuals of the same species in a given area at the same time. So the study of inheritance is a modern branch of science, and this began in the mid-19th century with the work of Gregor Mendel. So he was a monk who bred tea plants, and if, of course, you can look here with me, you'll see a depiction of such. Also see in your textbook, page 229, for another picture of tea plants. So Mendel was the first scientist to effectively apply quantitative methods to study of inheritance. So he did not merely describe his observations, he planned his experiments carefully, recorded the data, and analyzed the results mathematically. Although unappreciated during his lifetime, his work was rediscovered in 1900. During the decades following the rediscovery of Mendel's findings, geneticists extended Mendel's principles by correlating the transmission of genetic information from generation to generation with the behavior of chromosomes during meiosis. By studying a variety of organisms, geneticists verified Mendel's findings and added a growing list of so-called exceptions to his principles. Geneticists not only study the transmission of genes, but also the transmission of genetic information. As you will see in this chapter and those that follow, understanding the relationships between an organism's genetic information and its characteristics has become increasingly sophisticated as biologists have learned more about the orderly transmission of information in cells from one generation to another. So let us begin now with Mendel's principles of inheritance. So as we get here, just keep in mind there are a number of terms you all need to know and should be able to master. And I say mastery of these terms will ensure understanding of the content presented in the chapter. So if you cannot understand what I'm presenting now, please make sure you reach out and let me know so I can help you. But I would say the major part or the major component of this is to know these terms that I'll be using. So let us begin. So with this, Mendel was not the first plant breeder. At the time he began his work, breeders had long recognized the existence of hybrid plants and animals, meaning those are the offspring of two genetically dissimilar plants, or two genetically dissimilar parents, So when Mendel began his breeding experiments in 1856, two main concepts about inheritance were widely accepted. Firstly, all hybrid plants that are the offspring of genetically pure or pure breeding parents are similar in appearance. That's one thing. Secondly, when these hybrids mate with each other, they do not breed true, in that their offspring show a mixture of traits. Some look like their parents, and some have features like those of their grandparents. So Mendel's genius lay in his ability to recognize a pattern in the way the parental traits reappear in the offspring of hybrids. Before Mendel, no one had categorized and counted the offspring and analyzed these regular patterns over several generations to the extent that he did. Just as geneticists do today, Mendel chose the organism for his experiments very carefully. He chose the garden pea, Pisum sativum, and in using this garden pea, what he saw is that there were several advantages. So pea plants are easy to grow, and many varieties were commercially available. And another advantage is that controlled pollinations are relatively easy to conduct, in that the pea flowers have both male and female flower parts and naturally self-pollinate. However, the anthers, which of course would be that male part of the flower that produces pollen, can be removed to prevent self-fertilization. So pollen from a different source can then be applied to the stigma, that is that receptive surface on the carpel, or that female portion. So pea flowers are easily protected from other sources of pollen because the petals completely enclose the reproductive structures. So with all of this that has been stated, let's get into a bit more information about where we are now. So if you look with me, class, I'm on page 230, and I'm going to go through some terms. And these terms, class, are going to be helpful and terms you need to know as far as performing well on the upcoming test. Let us begin. So I've already gotten to terms such as heredity, terms such as genetics. So the first term now is going to be phenotype, and that is nothing more than that physical appearance of an organism. So there are many different phenotypes, and we'll get to a number of these, I would say, in lab and just all throughout the lecture. So you could say phenotype would be that physical appearance, such as a person having the widow's peak would be, of course, something that is a phenotype that you can see, or having mid-digital hair. So if you look at your hand, your right hand or your left hand, and look at your knuckle, just beneath that knuckle going towards your fingertip, class, there may or may not be mid-digital hair that's there. And there are a number of other things I could go over right now, but we'll save those, of course, for lab. Next up is genotype, and that is the genetic makeup of an organism. So with that, I'll just give you an example now. It could very well be that this, excuse me, I don't know why that took my H off to left field. I don't know why it took my allele to left field, but here we go. So this is an example of a genotype, and this is typically how it's done as far as teaching purposes. And I'll give you this would also be an example of a genotype. So those are two examples there. So having given those two examples, what I'll also do is describe those examples a bit more specifically. So having done those two, I'll call each of those genotypes, and I would say the genotype we see here is homozygous dominant. And I'm taking my time here because these things, class, are important to know. So I say homozygous dominant because there are two, and I'm going to draw here the top left-hand corner, two alleles. So that is an allele, that H, so to speak. And the word I'm using, class, is allele. I'm going to change it back to the black color here. That's A-L-L-E-L-E, allele. So that H, class, here is a dominant allele. And then I'll draw yet another H. I'll draw that here. And that, class, is a recessive allele. So you've now, of course, had an example here given, class, at the bottom of the screen with two dominant alleles, and I said that this is homozygous dominant. So now that I've done that, what I'll do next in class is move over to my OR statement, and this that is here is known as being heterozygous, or a hybrid, so to speak. So you have one dominant allele and one recessive allele. So those are two examples, class, showing you genotypes. Genotypes. Now, let's move on to what's next. So it states that true breeding line produces only offspring that express the same phenotype generation after generation. So I know there's a lot on the screen at the moment, class, but I'll make it make a bit more sense here. So that means that this class shows true breeding offspring, in that they would only express that same phenotype, class, generation after generation. So in his experiments, he crossed plants from two true breeding lines with different phenotypes, and I'll get more to that class a bit later. So from here, class, this mentions generation. So as far as generation is concerned, the P generation is that parental generation, and they are genetically pure individuals. So immediately thereafter, we get to that F1 generation, and that F class, that term F is known as filial generation. So I'll just put in parentheses F-I-L-I-A-L, and I said filial generation. So this is from the Latin for sons and daughters. So the F1 generation, or that first filial generation, is that first generation of offspring. And in his case, in his experiments, they all looked alike and resembled one of the two parents. In that second, or that F2 generation, that filial generation, the second generation resulted from a cross between F1 individuals or by self-pollination of F1 individuals. So of course we go from that known as the P generation, as you see in your textbook class on page 231, that's figure 11.3. So you go from the P generation to the F1 generation, and then from the F1 generation class thereafter to the F2 generation. So having gone through these, what we'll nextly get to is that very same figure. So here we have those F1 hybrids, and this is where one trait from a parent was masked by a trait from the other parent. So when I'm stating this, I'm just looking at what you have right here. And it says it's masked because what we actually have is that there is a dominant allele and a recessive allele. With that dominant allele and that recessive allele, we have this hybrid. And I don't want to do too much at this moment, but given that, there is a bit of masking in that you really don't see, class, anything from that recessive allele yet. And I'll make this make a lot more sense. All right. So given all of this, let us get to a few more terms, and I'm quite confident that all of this will make sense. So here we look at table 11.1, and table 11.1 provides these experimental results for 7 characters, and let's get to what a character is now. So a character is nothing more, class, than the attribute for which heritable differences or traits are known. Now keep this in mind as we get further into the lecture. And I'm pausing a bit because this is given, class. However, it's just not written out as, I guess I'll say, as bluntly as I'm giving it. So again, I say the term character, to ensure you're understanding where we're going, is nothing more than just something such as seed color. And maybe it's seed shape, or even pod shape, pod color, or even flower color. So those are all examples of characters for which there are heritable differences, or traits that are known for those differences that exist. So I won't spend so much time, class, here with his results. What I will spend more time with, class, are, I guess I'll go with Mendel's model of inheritance, and then we'll get back to some other terms. So you'll find his model of inheritance, class, on page 232, table 11.2. So he stated, class, there are alternative forms for a factor to account for variations in inherited traits. So alternative forms. So when he said that there are alternative forms, class, this is the term I gave you all moments ago, which is right here in the center where it says allele. So let's go back to where he mentions that. So I'll write it again, class. A-L-L-E-L-E. The term allele, class, I say is an alternative form of a gene. That's how I'll define it because that's how it's known now. So in that, I'll write here at the bottom, class, an allele, class, could be either this, that's a letter D as in dog, or that, and that is also the letter D in dog. So notice I said alternative forms of a gene. So the allele, class, here on the left is dominant. In this case, I'll say it's dominant. And the alternative form of the gene, class, here on the right, so this allele that's on the right is recessive. It's recessive. So given each of those, class, meaning having this allele on the left being dominant and that allele, class, on the right being recessive, it may very well be that the allele on the left, class, makes this, I guess we'll say, what do I want to call it? I guess I'll go with dark and light colored coats. So the allele on the left, class, would cause the coat, class, to be dark in color. Notice, class, that dark color, well, you help me, class. Is that a phenotype or is that a genotype, being dark in color? That would be a phenotype. And the allele, class, on the right would make that light in color. In other words, if their class were to be a genotype that looks like this, or a genotype, class, that looks like that, it would be dark in color, meaning homozygous dominant or heterozygous. However, the genotype, class, here on the right, being homozygous recessive, would be the only way you get that recessive phenotype being light. So let's continue, class, with that second model of inheritance. So inherited traits pass from parents to offspring as unmodified factors. And as I say unmodified factors, I just mean that Mendel did not observe offspring with anything that was intermediate. And there are exceptions here, in which, of course, there's a budding effect, and we'll get to those a bit later. At number 3, each individual has 2 sets of factors, one of each pair inherited from the mother and the other from the father. So as I've drawn here, class, at the bottom of the screen, I'll just accent it with this. In inheritance, class, there is one allele that comes from the mother and one allele that comes from the father. So in this case, maybe that came from the mother, and maybe this came from the father. And that, class, happens. It has to occur. That's what happens. So nextly, class, we'll go to number 4. The paired factors, and I'll call these the paired alleles, they separate prior to the formation of those reproductive cells. And this gets to that law of segregation, or that principle of segregation, which was presented last chapter as we had gotten to meiosis. In other words, we begin, class, with that diploid cell in that process, a diploid cell having 2 complete sets of chromosomes. Thereafter, there is that reduction division, class, where you go from diploid to haploid, and then thereafter, class, we get one more division in which we get the gametes, and yet still haploid. What I'm getting at is within those cells, let's just go to a different color, and let's go to, I guess, I'll go to this color here. So what happens is we have to go through replication, in which it replicates, class, the DNA. So that's why there are homologous chromosomes, and then, of course, we end up with this in those cells. I'm trying to make this something that can be read easily. However, my penmanship class isn't the best of which. So let me make it look better. There we go. So what I'm getting at is, next up, we get this. And this class happened all because of the law of segregation, or because of segregation as it was noted. I don't know how that changed my presentation and where we were in the presentation. I was using the eraser and trying to make this look a lot neater for you all. So that's what is meant, class, here in number 4, that those paired alleles separate prior to the formation of those reproductive cells. So there it happens. They must segregate themselves before you get those reproductive cells there. And they're all haploid, having a haploid set of chromosomes. Next up at 5, factors must be expressed or hidden in a given generation, but they are never lost. And I'll use a different color to make sure this makes more sense. And I'm saying it this way. So maybe the genotype class is this, that, meaning heterozygous. So what it's stating is it may be this that is expressed. However, that class has not been lost. It's still there. However, what is expressed would be that dominant phenotype. And that recessive phenotype just has not been lost. Lastly, at number 6, each factor is passed on to the next generation independently from all other factors. So this, of course, gets back to independent assortment. So it's not that they are passed on, class, together as one unit. So from here, I'll just move back to what are called genes. So genes, class, are units of heredity. And these units of heredity, they affect an organism's traits. And to make that make the most sense, class, it's right here. In other words, class, a gene is a DNA sequence. In other words, a sequence, class, of A's, T's, G's, and C's. As you all learned about DNA, or at least we'll learn a lot more about DNA, class, coming up in the next chapter. So it's that a DNA sequence, class in a chromosome contains this information. And from here, of course, it's to make an RNA or protein product with a specific function. So I'll do it as such. We all have known of DNA in some respect, called deoxyribonucleic acid. So what happens is, and I'm getting to what's been, class, said to be that central dogma, the central dogma of DNA, in that we begin with DNA. And the way in which, class, we all, I guess you say, get more DNA is by the process of replication. DNA, of course, is replicated to, of course, get more DNA. Thereafter, the process of which we go, class, from DNA to RNA. So that process, class, is known as transcription. So DNA, class, is replicated, and now we get to the process of transcription. In other words, class, DNA is transcribed into RNA. I say again, DNA is transcribed into RNA, and ultimately, class, RNA is translated into a protein. So all right, here's the process of translation. So to make this make more sense, this, class, is that central dogma of DNA, in that these genes, and all genes are encoded, class. All genes are within our, of course, DNA. So what happens is DNA is replicating, class, all genes for, of course, all of the cells. And not all of our cells are alike, so it may be that in this cell, these genes are turned on. However, in that cell, those genes are turned on. All of our cells do not, class, have all genes turned on. Because, of course, our cells are different. They are specific. Nextly, class, I said DNA is transcribed into RNA, which is ribonucleic acid, and then RNA is translated, class, into the protein. That, class, should give you, I say, everything needed to understand what we're doing here at this moment. So the next term up, class, is the term locus. So a locus is nothing more than the location of a gene on a chromosome. And I'll draw this briefly. I think I'm still in the blue color. Yeah, that's blue. So let's draw this, and then I'll draw that structure there, and then I'll draw that. So this is just a large chromosome. I drew it large so you all could see it quite well. And then what I'll do here is I'm going to just draw that, because I'll call that, where I have drawn the orange, class, the locus, the location, class. I'll repeat, the location of a gene on a chromosome. So the site a gene occupies on the chromosome. So of course, I've gotten to alleles already, meaning that they are different forms, an alternative form, class, of a gene. And then keep in mind that those alleles, of course, they occupy a corresponding location. They occupy corresponding loci on homologous chromosomes. For instance, let's just say this is chromosome number. I guess I'll just jump off on number, what number? Let's say chromosome number 7. Since this is chromosome number 7, it is also the case, class, that this too is chromosome number 7, looking like that, and then looking like this. And I hope you all are following me with that there. And then, just to make this interesting, what I'll do now, class, is state that maybe we have this allele at, wow, that does not look like the letter A is an apple. And I was stating that maybe we have this allele at the locus there on that chromosome 7, and maybe we have that allele, goodness, that should be a recessive A, but it doesn't look recessive, you all. So let me make this look recessive. And maybe we have that allele there. In other words, class, we have this heterozygous genotype at this locus, meaning the loci, they correspond because those are two chromosome 7. And to go back and review, one of those chromosome 7's, class, came from the father, and one of those chromosome 7's, class, came from the mother. And that, class, is how everything is determined. And yes, this is a simpler way of explaining it, but it's the very same way. And if, in fact, class, those two alleles were identical, this would, of course, be said to be homozygous, be it homozygous dominant or homozygous recessive. But in this case, class, it is heterozygous, not homozygous, meaning there are two different alleles on homologous chromosomes. So the next portion just states that during meiosis, those alleles, and I'm referring to all alleles, class, for each locus, they segregate from each other to ensure, class, variation, to give us those differences. And you can see, class, the very same thing shown here as I drew it, class, earlier in this lecture. So be sure, class, you review these figures and terms to ensure you understand what's happening. So, of course, we have the homologous chromosomes that are there. You all see at the loci those 4 differing alleles that, of course, make up those. And then, of course, there, and ultimately, class, there. And the very same thing is the case here, class, meaning for these loci and the color of fur. Next, we get to monohybrid and dihybrid crosses. So the monohybrid cross is showing, class, the inheritance of 2 different alleles of a single locus. For instance, let's just do this now to ensure you all can complete a monohybrid cross. So here we go. So using what we have here, let's just deal with, I guess I'll go with brown hair. Let's go with brown hair. And in this case, I'll call this the allele for brown hair, and I'll call this the allele for black hair. And these are going to be from the father, and these will be class from the mother. I'm referring to the gametes. So I'll place the father's first allele there, first allele there. So I hope you can state, class, that the father has what color hair? Brown hair. And I'll put these alleles here for the mother. And what alleles, class, what is the phenotype for the mother's hair? I hope you all say black. So in this respect, we'll get this, and to make sure we can all do this, that goes down, and that, class, goes to the right. That goes down, and this allele still, class, goes to the right. And of course, we go down again with that dominant B, and we still, class, go to the right with that recessive B. And then we go down with that dominant B, yet and still, and to the right again, class, with that recessive B. So we've now, class, completed this monohybrid cross. Now let's talk about the possibilities. So what would be, class, the genotype frequency for those possible combinations? And I said the genotype frequency. Genotype frequency. I'm going to abbreviate the term frequency. So I would say, class, it should be 100%, or at least 4 out of 4 heterozygous. So it would be that, and the frequency is 4 out of 4. In other words, class, for every time those individuals reproduce, they will always get what color hair? Or at least, they will always get, class, those genotypes that are heterozygous by crossing this pure brown with that pure black. Now let's talk about, class, the phenotype frequency. The phenotype frequency. So the phenotype frequency, class, would be what? 100%, or at least 4 out of 4. What color? Brown. So of course we would say they are all brown, and that would be, class, 4 out of 4 brown. So there you have it. You've now completed a monohybrid cross. And please ensure, class, that you all are prepared to do this on your test. So with the dihybrid cross, take yourselves, class, over to, I guess I'll go over to page 236. Page 236. On page 236, look at figure, class, 11.8, and that would be showing your dihybrid cross. And I'll try to draw this out for you all in a way that's understood. So that's there, that's there, that's there, that's there, that's there, and that is there. So with this, class, this comes directly from your text. So I'll use the very same alleles here, class, in our dihybrid cross, but keep in mind that the dihybrid cross, of course, are going to cross and show the inheritance of different alleles at 2 different loci. So in this case, I'll use that same allele that's here. However, in this case, class, I'll say this would make the individual black in this case. And that, class, will make the individual brown. Notice I'm changing what I just did quite moments ago. And in the very same way, I'll say that this has long hair. This would make long hair. And that class will make it have short hair. And I'm using the letter or the allele L to ensure I'm not writing class the letter S. I want you all to be able to tell what's going on. So I'll now add the gametes class. So these gametes that are going to be here are going to be class from the mother. So they are as follows. They're going to be that allele and that allele, that allele, and this allele. That going to be that allele and that allele, and then this allele and that allele. So for the father class, which will be over here, it will be this allele and that allele. It'll be that allele and this allele. It will then be that allele and that allele, and then two recessive alleles. And I'm doing this to make sure you all can complete a dihybrid cross class. So now, what you should do is the very same thing we've done in the past, in that we will then have two dominant alleles class, ensuring that there is black hair, and that black hair, as far as being long or short, will be black hair that is long. And I'm going to make sure I do my best to make this neat so that you all can reference this. So I'll continue now, class, with, of course, black hair that is also long. We still, class, have black hair because we have that dominant allele there. And this will be black hair that is long. And we will still, class, have black hair that is long. So those are possibilities. Yet and still, black hair that's long. Black hair that is now short. Black hair that is long. And then black hair that is short. Here we have black hair that is long. We have black hair that is long. We have now, class, brown hair that is long. We now have brown hair that is long. Here we have black hair that is long. We have black hair that is short. We have also here brown hair that is long. And then we have brown hair that is short. So you have done, class, your first dihybrid cross. So given this, class, you can go through, of course, what we've done. But as far as our phenotype frequency, I now say the phenotype frequency. As far as the phenotype frequency, you should count these, class, and I would say use check marks or use whatever use, but those that are black with short hair, class, there are 9 of those. So 9 black and short hair. So let's look at this again. There are 3 of those with black long hair. And then there are 3 of those that have brown and short hair. And then there is just one class with brown long hair. And let me step back and look at this one more time. So to make sure I say the right thing, class, I'm going to go back here. This dominant letter makes hair long. So it's actually 9 of those individuals that have black long hair. There is one here. 1, 2, 3, 4, 5, 6, 7, 8, 9 black long-haired individuals. Black long-haired individuals. Then there are black and short hair. So there is 1. There is 2. There is, where is it? 3, black short. And I'm making sure I do this with you all because I misspoke just by mistake. And next up, I'm going to use yellow, so it's going to get really light. So these individuals will be brown and short. So there is, brown and long. So there is 1 brown and long. There are 2 brown and long, and there are 3 brown and long individuals, making sure you can see my yellow. And then there is just class 1. There is just one class that has brown short hair. And that's right here. So there's 1 with brown short hair. I'm glad I caught what I was saying incorrectly. So the Punnett Square was done correctly. I just used the incorrect name for a phenotype based on long and short hair. So on your test class, prepare yourself to, of course, complete monohybrid and dihybrid crosses. And there you have it, class. So let's move on down to the test cross. So a test cross, class, is done because sometimes there are alleles that are dominant and, of course, alleles that are recessive, but you cannot tell, class, based on phenotype, the organism's allele. So the way to determine such, class, is to perform a test cross, and that just means that you would cross, class, that individual with that unknown genotype with a recessive individual to determine, class, that unknown genotype. So from here, class, I'm going to keep on rocking, and of course we've got to our dihybrid cross already. But just keep in mind, class, that the dihybrid cross was done because we had 2 different alleles at different loci. So non-homologous chromosomes. So we've done this already, class. So I'm now moving on. So with the principle, class, of independent assortment, we did this already, but to ensure you get it, class, this just means that members of any gene pair segregate from each other independently of other gene pairs. And this, class, gives us genetic recombination or genetic variability. The same, class, was the case with segregation. So with segregation, that just means that these alleles separate themselves independently of each other, and this happens, of course, during meiosis. 2 ways in which, class, we get what I call variation. So as you see it, it's not together that any of these alleles, class, separate themselves. That's why it's called what it's called, being independent, class, assortment. So with probability, class, I won't spend much time here because we have a lab for this in which we'll get to probability. But just keep in mind that mathematically, what's occurring, class, does not depend on what has already occurred. It's all, class, random. So now let's get to the inheritance and chromosomes. So with this, we'll begin with linked genes. So this came from Morgan here, Thomas Morgan Hunt, or Thomas Hunt Morgan, excuse me. So in this, that he demonstrated that the genes that are inherited class in a linear pattern on each chromosome. And what I'm saying is, with linked genes, I'll be blunt class. Linked genes are typical to be inherited class as a unit. So inherit together. So I can't say it any way other than that. So if they're together, they'll be inherited together. And to ensure that you can get what I'm saying here, class, I'm going to go to something you all, I would say, know a bit about. And we're going to get to those sex chromosomes. But to make sure we get there and get there the right way, class, just know that here in humans, class, we, of course, have 23 pairs of chromosomes for a grand total, class, of 46 chromosomes. And I'll say that we are 2n equals 46, meaning our diploid number, class, is 46, and it's because we have 23 pairs of chromosomes. So note this for your test, because, well, this is something that you all just should know. So with that, we have 22 pairs of autosomes and 1 pair of sex chromosomes, giving us that grand total of 46 chromosomes. And that's that 23rd pair, class, that determines our sex, determines your sex. So, of course, Huff being a male, my 23rd pair, class, would be that X and the Y, and XY. In other words, females have 2 X chromosomes, males have 2, of course, being X and Y. So one of each. We are heterologous, if you would like to say it that way. Heterologous. All right, so having gotten to that, let's keep on going to where I really would like to get to ensure you understand what's happening here. So during the evolution of that X and Y chromosome, most of the original functional genes were lost from the Y. That Y chromosome, class, is actually quite small. It's a great bit, class, shorter than the X chromosome. Alrighty then. So from here, let's get to the function of the X and Y briefly. So with the function of the X and Y, just keep in mind that the X chromosome contains important genes that are not necessarily related, class, to sex determination, and they're required for both sexes. Notice I said males have 1 X and 1 Y. However, it's that Y chromosome that determines the male sex in mammals. So the way in which it happens, I say, class, is by way of what is known as that SRY gene on that Y chromosome. So that SRY gene on the Y chromosome is that genetic switch, which causes the testis to develop, or the testes, excuse me, to develop. You got to have 2 testes. Being born with a testis, well, get to it. So it requires, of course, that SRY for that genetic switch, class, that then causes the testes to develop in the fetus. So the testes then, of course, would begin to secrete the hormone, class, known as testosterone, which you also on your test, which causes other male characteristics to develop. So I hope this is making better sense now that I've done this with you. All right, but just keep in mind, class, that today about 95% of that Y chromosome is male specific. So either the Y chromosomes retain certain genes that code for proteins and that determine maleness, or perhaps the mutations occurred in the existing genes on that Y chromosome that made it, class, that male determining chromosome. So with this being the case, I guess I'll just go to what happens next. And this, class, is from your text. You'll find this on, well, now that I think a third time, you won't find this, class, given in your text, but this just gives you representative examples of sex determination in animals. So, of course, this is in humans, yet again in humans, and of course in humans, definitely in humans. And then it gets, class, to a few cases, class, where it is not in humans. So what I was trying to get to, class, moments ago is right here before you, being X-linked genes. And you may have even been taught that these are called, class, sex-linked sex-linked genes. However it was taught, class, let's get to it now. So with this, it says that a female receives X-linked genes from both parents. And of course, if it happens to be, of course, from that male, it's going to be that a male receives all his X-linked genes from the mother. In other words, class, if in fact the male, class, has received some X-linked genes, it's of course going back to his mother. And I'll make it make a bit more sense now. In other words, class, in males, class, they have an X and they have a Y chromosome. Males do. A Y chromosome, class, only comes from the male. Hence I said, a male that receives some X-linked gene receive that X-linked gene, class, from his mother, because his father, which provided, class, that Y chromosome, did not provide, class, that X-linked gene. So it states, class, being hemizygous, meaning a male is neither homozygous nor heterozygous for his X-linked alleles. Instead, he is always hemizygous, meaning he has only one copy of each X-linked gene. In other words, class, if in fact someone has that, they will have that disorder because that male, class, has how many X chromosomes? If you can see, class, the top right of the screen, only one X chromosome. So if it's there, it will, class, be expressed in every case. So what I'm getting to, class, is that females receive an X chromosome from their mother and an X chromosome from their father. Males, class, receive their Y chromosome, I say again, only from their father, and the X chromosome will always and only, class, come from their mother. So I'll state again, all X-linked genes come from the mother, and every allele on the X chromosome, I repeat, is expressed, whether or not, class, the allele is dominant or recessive in the female parent. So let's go through a few examples here, and this will be, I say, make a lot more sense. So let's say that it's this and that. And let's just go with color blindness. So this, class, would have the female, in this case, have normal vision. This female would have normal vision. And I'm writing in red. Why, I do not know. I'm going to change this color up from red. I just can't do this red. So next up, class, let's look at this woman. And I'll say she has that with this. So she still, class, has normal vision. She still has normal vision. However, class, she is now a woman with normal vision that's also stated to be a carrier. And she's a carrier because of the presence of that recessive allele there. So let's go to males. So with males, I will put it as being, I guess I'll stay with my color that is called green. And let's say he inherited, and I'll go to this. So I'll ask, class, what type of vision would he have? Would he have normal vision, or would he have, class, color blindness? He, class, has what? Normal vision or color blindness? He, class, has normal vision, and it's because he has that dominant allele inherited, class, from his mother. Lastly, of which, class, let's look at this male. So this male, and I guess I'll go to this color here. Let's go there, and provide allele here. And help me out, class. What type of vision would that male have? that male class would indeed be color blind. And it's all because he inherited that recessive allele from his mother. So, with that being the case, let's get to an example you all can work out here. So let's cross a normal male with a heterozygous female. So to help you out, class, that will provide person with normal vision, and this class will make the person color blind. And it's not that this is being made up, class. Color blindness is an X-linked disorder. So this example is nothing that I'm just pulling out of thick or thin air, if that makes a bit of sense. All right, so I'm just trying to find a better color to write with you all. So let's cross, I said, a woman class. Wow, I don't know why this computer does that. So let's cross a woman that is heterozygous. So she has this here. She's heterozygous, i.e., also known as a what, you all? Carrier with a male that is a male with normal vision. So I'll now draw the Punnett Square like you all should draw your Punnett Square. I'm just going to change the color a bit. So let's go to Punnett Square. It looks like this. And then I'll draw that there before I forget. I'll put the female at the top right. So she'll go here. And I'll put him there. And now let's add the gametes. So she provides either this. Wow, this, I have got to find out why it does that. I don't know why it does it. So I'll put this here. And I'll put that there because she is a carrier. Then I'll put this here. And I'll put that. So let's fill out the Punnett Square. So I'll bring each of those down, and then I'll bring each of these to the right from him. Then I'll bring down and over. So I'll go over with this. And then I'll go down with that. And then I'll go down with this. And over with that. And then I'll go down with this. And I'll go over with that. So if, in fact, class, you're given this on the test, and I'll say prepare yourselves for it, what would be the probability, class, of them having a son that is colorblind? What do you all say the probability is? If you can't see it, I'll just tell you, class. For every time that they have a child, I repeat, for every time that these two individuals have a child, there is a 1/4 or 25% chance that they will have a son that is color blind. I said again, for each time they have a child, there is a 25% chance that that child will be a boy with color blindness. And this is why in every instance, class. And it's random. It's not that they have a son the first time that's colorblind, that they will not or will the next time. There's that 25% chance for each time, class. So I hope this helps with sex-linked disorders. And the very same, class, is the case for hemophilia. And it's also an example, class, of a sex-linked disorder. If you have questions about this, class, I would definitely say please email me. Please ask. Because I'll say I will look for you all to know exactly this and how to work out a problem as such on your test. Back to where we left off. So in that carriers class, I hope this is understood. But that just means that if in fact she is a carrier, that means that she has class an allele that is recessive class on one of her X chromosomes to be a carrier. All right. So we've already done an example class. So let's get nextly to extensions. Extensions of Mendelian genetics. And in these extensions class, just keep in mind that dominance is not always complete. There are class examples that are incomplete dominance and even co-dominance. So with incomplete dominance, it states that the heterozygote has a phenotype that is an intermediate, meaning that intermediate phenotype. So this is where we class get colors such as pink. So in this case class, I guess I'll rock it out this way. Let's cross what is known as, I guess I'll go to flowers, and I guess I'll use the example given. So let's begin class here at the P generation. At this P generation class, we have a red flower and a white flower. So by taking that red flower and that white flower, of course, in that F1 generation, what we get class is all pink flowers. And it's that because the heterozygote expresses that intermediate phenotype. If you don't know what I mean, class, this is what I mean right there. That class is the heterozygote. Yes, there are 1s and 2s, but since class it's R1, R2, that is a heterozygote. It's not R11. If it were to be R11 class, the flower would be red. If it were class R2, R2, the flower would be white. So just know, class, that's why I say the heterozygote expresses the intermediate phenotype. So let's cross class what we have here as being 2 pink flowers. So when you cross class 2 pink flowers, what we get class is this 1 to 2 to 1 ratio, meaning 1 red flower, 2 pink flowers, and 1 white flower. So that's how we get class, the 1 to 2 to 1 ratio. And I wrote that out that way, class, because 1/4 class red, half of them class white, and one class being, half of them pink, and then 1 flower being white. That's why I have that 1 to 2 to 1 ratio. Up here nextly, class, we get to co-dominance. So in co-dominance, class, is this defined? Yes, it is. In co-dominance, the heterozygote simultaneously expresses each of those phenotypes, or both phenotypes, class, of the homozygotes. So in this example, class, it's, of course, ABO blood grouping, or ABO blood types. So here, class, types A, type B, types AB, and type O are controlled by 3 alleles, class. I'm going to write it right here. They're controlled by 3 alleles at a single locus. At a single locus. So that's why I say, class, that this is an example of co-dominance. But of course, put somewhere on your paper, class, that this is also an example of multiple alleles. For instance, I'm going to write it now too. This is also an example, class, of multiple alleles. And I say multiple alleles because, class, there are 3 alleles that give us, class, the phenotypes you all know as being your blood types. So let's get to it. In this case, class, the phenotype type A blood. The phenotype A blood. The only way on earth, and go to red since we're referring to blood, the only way, class, you get type A blood is by being, class, either IA, or being, class, IA, I. And that's a recessive I. Class, I would say again, this is the only way on earth to get what's called, class, type A blood. Either genotype class IA, or IAI. So to spare you class the rest of such, I'll use the chart as given. To get type B blood, it's quite similar in that you have to have IB, or IBI. That gives you class type B blood. So I don't know what your textbook class is doing here for type AB blood, so I'm glad I'm able to write this out for you. So to get type AB blood, there's only one way class to get that. In that you have to have IA, IB class, and this is the only way for a person on earth class to have type AB blood. Nextly class is for type O blood. And for type O blood, its only class going to be II, meaning such as myself. I have type O blood, so that means I have two recessive I's class to give me type O blood. So I won't speak so much class to surface antigens or antibodies, but let's spend a bit of time here since it is presented class, because I'm sure someone may wonder, what in the world is that, or what do you mean? So if a person class has type A blood, that means they have type A, or at least surface antigens A. A person class with type B blood have antigens B, and a person that has type AB blood have both class antigens A and B. And for myself, class with type O blood, I have neither of those two antigens. Next up, class, I'll state that let's go to antibodies within the serum. So if you have type A blood, you have type B antibodies, or anti-B. If you have type B blood, you would then have class antibody A, or anti-A antibodies. And then, of course, if you have type AB blood class, you have neither of those two antibodies in your blood. I repeat, you have neither of those two antibodies in your blood. And of course, for someone such as myself, class of type O blood, I have both antibodies in my blood. So anti-A and anti-B are in the blood class of me. So with this being the case, blood is needed class. If you can, I would say donate. I say if you can, please donate. But please also do not get alarmed. In other words, using the information class given here, a person could most definitely find out whether or not class that he is or he is not, or she is not class the child of that mother or father. So please be careful with this information. And I cannot stress enough, please be careful with this information. In other words, if both parents, I repeat, if both parents were to have type, I repeat, if both parents would have type AB blood, and you yourself, class, have type O blood, I'll leave it up to you as far as being whether or not you are the child of parents of such. And the very same class would be the case now. If in fact both parents would have, of course, one have type A blood and one have type B blood, is there a way class of a child with type O blood? You're dang right. That means that, of course, both of which, and there is a mistake, I've just now noticed, class, this is incorrect. In other words, if both parents were respectively to have type A and type B blood, this would be, of course, the genotype that they would have to have a child class with, of course, type O blood, because that child would, of course, had to have gotten class the recessive I from each parent to, of course, have the type O blood. That's why I stated, if in fact you have type O blood and each parent class has type AB blood, I'll hush. All right. So what I'll nextly get to, class, and I've already gotten to multiple alleles. And to make sure you understand what I stated, class, that there are, of course, more than just, of course, two alleles. There are 3 alleles, class, at one locus. So to get to where we're going, class, with pleiotropy, it's just the ability of one gene to have several effects on many characters. And as I speak to pleiotropy, class, it's the case, class, meaning can be traced to that single fundamental cause. So it might be a defective enzyme that affects, class, many types of cells. And in this case, it's evident in many genetic diseases. For example, if someone is homozygous for the recessive allele that causes cystic fibrosis, where they produce abnormally thick mucus in many parts of the body, including the respiratory, digestive, and reproductive systems. So it's class that multiple effects it's had there. So next up with epistasis. So it's a common type of gene interaction in which the presence of certain alleles of one locus can prevent or mask the expression of alleles at, of course, of different locus, or even express their own phenotype instead. So epistasis class, it just means it stands on its own. So coat color in Labrador retrievers is an example of epistasis, and it involves a gene for pigment and a gene for depositing, of course, color in the coat. So two alleles for the pigment gene are, of course, B for black coat, and its recessive counterpart, of course, is that lowercase b, or that recessive b, for brown coat. And then, of course, it can be epistatic class and block the expression of either of those, in that, of course, you get a yellow coat, regardless of the combination class of those B alleles. So you can see class, in this case, the gene interaction. So the black labs have genotype B and, of course, E, meaning dominant B and dominant E. Chocolate labs class have genotype 2 recessive Bs and the dominant E. And the yellow lab class would have, of course, a dominant B and 2 recessive Es, or just all recessive Bs and Es. It's quite interesting, class. Thanks a lot, epistasis. So next we get class 2, polygenetic inheritance. So with polygenetic inheritance class, the offspring exhibit continuous variation in phenotypes. So it's alleles at several different loci that affect each character. So with skin color class, and it's called Homo sapiens class, there are as many as 60 loci that account for skin pigmentation. So I'll make this, I guess I'll say brief class, but to keep things simple, I'll just use 6 different loci with letters A, B, and C. So the capital letters class are, of course, incompletely dominant alleles, producing dark skin. So the more capital letters there are class, the darker the skin, because the alleles that affect skin pigmentation in an additive fashion. And let's get to that now. And if this doesn't make sense, please ask. So I'll draw class, maybe what you've seen in many of your math classes. Just this that is looks just like that. All right. So on such, let's just go here. And I'll begin kind of in the center to find my, well, before I do that, let's go to labeling my axes. So to label my axes class, here it will be skin pigmentation. This is skin pigmentation. And then on my y-axis, it's number of individuals in my F2. So it's number of individuals, and it's in that F2 generation. So what I'm getting at is the F2 offspring would, of course, be what I'm working with here. And as I do this, just keep in mind, class, that I would be crossing 2 heterozygotes. In other words, class, I'd be crossing that person that is this, that, and then this, that, and crossing a person, class, that is also this, that, this, and this, that. All right. So as we get those possible combinations, we have this. So in the approximate, well, here, class, this individual would be very dark here, and this individual will, of course, be very light. So let's begin now. So that would go there, and then here to there, and then there, 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 and then there, there, and then there, there. So what provides from this, or what we get from this class, is from such happening class, meaning as we have this called polygenetic inheritance, what we get is called continuous variation. And please pay the most attention, class, as we are here. I say continuous variation because it's that normal distribution for those of us who have had a bit of statistics before. If you haven't had statistics yet, it's okay. And I say continuous variation because we get this normal distribution curve, or this bell-shaped curve. And it's, I'll be blunt, class, it's not black and white. It will never be black and white. And the reason being is because these are not discrete characters. So let me write up here continuous. It's because of this continuous variation. Continuous variation. And I just said never black and white because, of course, that was black and white, class, when we got to the discrete characters. With discrete characters, that was what was experienced, class, with complete dominance. It was either the pea plant was tall or short. It was those seeds, class, within that pod were either green or yellow, round or wrinkled. Well, with polygenetic inheritance, class, and all of these alleles, class, meaning at some 60 different loci, things are never just black and white, class. And you could extrapolate this, class, to, I would say, the entire human population here on Earth. And you're saying, well, what do you mean? Well, what I'm saying is, I guess this is that approximate center, class, of our bell curve or our normal distribution. And I'm speaking, class, to statistics right now. Most of which, class, I repeat, most people on Earth are going to be within 2 standard deviations, class, of the mean. And the mean, class, is of course denoted by mu, or this intermediate here, this middle. And that, in fact, class, I would say there are very few individuals, class, on Earth that are albino. And you can likely count on one hand those number of individuals you've seen in your life who are albino. And the same, class, could be said about those individuals that are this dark. And it's all based on this. And this is regular old statistics, class, and nothing to be afraid of. But this is also going right back to biology. So I hope this makes better sense. And if it doesn't, just, of course, ask. Just ask. So from here, class, we'll kind of end things, going back to where we just were. Genes, class, interact with the environment to, of course, shape phenotype. For instance, the hydrangea, which I love hydrangeas, class. Hydrangeas are all the same phenotype. However, class, their flowers vary in color depending upon the availability of aluminum, which in turn is dependent upon soil pH. So blue flowers, class, are present with pHs from approximately 5.5 or less, and purple flowers, class, are going to be shown in a soil with pH class of 5.6 to 6.4, and then, class, pink flowers from a soil pH of 6.5 to 7.0. So it's all because of, class, the biology and those environmental factors. So this, class, has been your instructor, Scholar Huff. This has been Chapter 11. Study it well, class. Take great notes. And if you need help, ask, reach out, come by my office, call me, schedule an appointment, or send me class before or even after lab. Thank you all for listening, and prepare for class well.